Now coming to muscle physiology, the principal unit is sarcomere, and sarcomere is the distance between two Z lines. So distance between two Z line is sarcomere. Then we have thin filaments attaching to these Z lines and the thick filaments attaching to these Z lines. The thick filament in between these Z lines. So these are the thick filaments and these are the thin filaments. This is the structure of a sarcomere. So at rest, these are the Z lines, and the distance between two Z line is one sarcomere, and you can see the thin filaments. These are attaching the Z lines, and we also have thick filaments. So these are the thick filaments that is in between the thin filaments both sides. Now the distance between two Z line is one sarcomere, and we have the distance or the width of the thick filament. This width is known as A band. So this width is known as A band, and one full sarcomere composed of two half I bands. So this is the I band, this one, the whole thin filament. So this is the whole thin filament. The thin filament is isotropic to the polarized light. Therefore, it is I band. A band is anisotropic to the polarized light. So the thick filament is A band, complete thin filament is I band. And one sarcomere, which is the distance between two Z line, this is half side this I band, half this I band, and we have one complete A band. So a complete sarcomere is one complete A band plus two half I bands. Now the space that is present between the I band, we have this space in which we have only A band. This space is known as H zone. So H zone. Now if you see in our picture also, diagram, we can see this is the, let's redraw again. So these are the thin filaments on either side of the Z line. So thin filaments on either side of the Z line. Now this is the complete I band, and here we have a complete A band. So this is a complete A band which constitute of thick filament, and I band which constitute of thin filament. Now the area or the space that is present between this thin filament, which constitute only the thick filament, this zone is known as H zone. This is H zone, and in between this, in the center of this H zone, in the center of the H zone at the thick filament, we have a dark line which is known as M line. Middle line, you can remember, it is present in the middle of this M line. This is the M line which is present on the middle of the thick filament. So that is a complete sarcomere. Now this is a two-dimensional structure. If you see the cross-section view of a Z line, so Z line is hexagonal and it is surrounded by six thin filament. So these are the thick filaments surrounding the Z line. and we have thin filaments. These are the thin filaments, thin one. So thin Z line composed of, or it is surrounded by 6 thin filaments and one thin filament. If you see one, this is one thin filament. This one thin filament, it is surrounded by 3 thick filaments. So if you take this one thin filament, it is surrounded by 3 thick filaments. So thin to thick ratio for the filament is 2:1. Now what happens? The initial, there is a law which is known as Starling's law, which states that force of contraction is directly proportional to the initial length of the muscle fiber. So the length is directly, if the muscle is longer, the force of contraction is going to be longer, and this Starling's law is applicable for skeletal as well as cardiac muscle. Now, greater is the initial length of the muscle. So if I say a normal resting length of a muscle, if it is 2.2 microns, so the normal length, which is length of the muscle at the rest, is 2 microns. And according to the Starling law, greater the initial length of the muscle, greater is or stronger is going to be the force of contraction. So once you start putting weight or pressure onto the muscle, then once the muscles start contracting, there is going to be increase in the, so once you start putting pressure, there is going to be increase in the length of the muscle. So from 2 microns, which is at the rest, it goes from 2.1, 2.2. Likewise, at point 2.2, there is going to be stronger contraction forces. This length produces the maximum contraction forces, and this length is known as optimum length. Then we have a capacity of our muscles to stretch. So this muscle can stretch up to, say, 3.7 microns. So that is the maximum length of the muscle can stretch. So this is the maximum length it can stretch. If the length is more, that means there are stronger forces of contraction. That is according to the Starling law. Now, we have 2 proteins that are attached to this sarcomere, or that are present in the sarcomere. So we have a titin protein, which is coming from a Z line and which is present between the thick filament and going to the another Z line. So it is anchoring the thick filament into the with the Z line. So this is the reason that thick filament is suspended in its position. So this titin, it is a protein. In fact, titin is the human largest protein. It is anchoring the Z line to the thick filament, and the function of this titin protein is the alignment of thick filament to the sarcomere. If there is defect in this titin protein, the disease is known as limb girdle muscular dystrophy. There is another protein which is known as dystrophin protein. So dystrophin protein is present between the thin filament. So it is connecting the thin filament to the sarcolemma. Here we have sarcolemma. So in the sarcolemma we have sarcoglycan protein, and we also have dystroglycan protein. So this is the beta subunit of dystroglycan, alpha subunit. This side is the alpha subunit of the dystoglycan protein, the beta subunit of the dystoglycan protein. So this is dystoglycan protein. Then this also in the sarcolemma, we have sarcoglycan protein. So there are 4 subunit of the sarcoglycan protein, which is alpha, beta, delta and gamma subunit of sarcoglycan protein. So this dystrophin protein, it is attached or linked to the thin filament, and it reaches up to the sarcolemma. Now in the sarcolemma, this dystrophin protein is attached to the beta unit of the dystoglycan in the sarcolemma, and in between it is linked to the alpha dystoglycan also. So this beta is attached to the alpha dystoglycan, but this dystrophin is attaching the thin filament to the beta dystoglycan. So two dystoglycans linked to 4 sarcoglycans. There are number of total 6 glycans in the sarcolemma. Now the tension which is generated by the muscle, by the sarcomere, which is generated by the actin-myosin interaction, it is transported exteriorly to the membrane, to the sarcolemma, and later to the laminin protein. So this transport of forces, transport of tension from the muscle to the membrane is because of the dystrophin protein. And if there is a defect in dystrophin protein, which is seen in case of Duchenne muscular dystrophy. Now if you see a sarcomere, we have I band, which constitutes of thin filament on either side of the Z line. This is the Z line. On either side of the Z line we have the thin filament. This thin filament, the whole band of the thin filament is known as I band. We also have A band. Now if you see, A band is the entire length of this thick filament, and in between this A band you can see there are also thin filaments that are present between the A band. So the A band constitute of thin filament as well as thick filaments. And we also have a H zone that is present at the center of the A band, and at the center of the H zone we have a M line, which is the middle of the thick filaments. So there is also a pseudo H zone that is seen on the either side during the muscle contraction. Now coming to muscle contraction. Muscle contraction is when the I band slide over each other. Now what happens? These Z line come closer. When these Z lines, say suppose these are the two Z lines normally in a zigzag manner, right? Now when they come closer, the I band are going to come and approximate to each other. The thin filament is going to approximate to each other, and there will be disappearance of this H zone which was present in the center when there was no I band. So the thin filament are going to attach to each other, and there is going to be disappearance of the H zone when the muscle contraction take place. So Z line come closer and A band is going to remain constant. So there is no change in the thick filament. The thick filament remains as it is, and the thin, because the Z line is coming closer, and you can see from the Z line these thin filaments are anchored to each other. Now the thin filaments are anchored to each other with the proteins, which we'll talk in a while. So the thin filament come closer and there is disappearance of the Z line. Also the length of the H and the I band also decrease because the Z line is coming closer, and the length of the A band remain constant. It doesn't change with the muscle contraction. So whole sarcomere shortened, I band shortened, A band remains unchanged, it remains constant because A band is the thick filament and thick filament is not attached to the Z line. Then we have H zone also, which disappears. Now when the H zone disappears, this line that is present in the center of the thick filament, it becomes very prominent. So the M line become prominent and H zone disappears. Now at the 1.5 micron contraction, now the muscle is going to contract, it is going to shorten in length. From 2 microns, when the muscle contraction is 1.5 micron, there is going to be appearance of M band which is present at the center and it becomes dark. We just now talked about the muscle contraction. Now this darkness of the M line, which is related to the contraction of the muscle, is known as CM band. So this CM band occurs at 1.5 micron, and when more contraction takes place at 1.25 micron contraction, there is appearance of a Z line because of the Z line coming closer to the thick filament. So whole of this space is vanished because of the muscle contraction, and now the thick filament is coming closer to the Z line, and there is CZ band that appears at 1.25 micron contraction. So the muscle contraction, when the muscle contracts, you can see this is the contraction. So the thin filaments, they come closer. There is disappearance of the H zone. There is no H zone. Then there is decrease in the length of the I zone. And we also have M line that becomes prominent. And the sarcomere also comes closer. Now earlier, this was the distance between two Z line. Here the distance between two Z line is shortened when the muscle contract. That is why when the muscle contract, earlier if this was the size of the muscle, when the muscle contract, it shrinks in size. So the whole sarcomere in the muscle, they shrink in size, and as a result of that, the muscle contraction or shortening of the muscle takes place. Now when we talk about thick filament, this is made up of 500 myosin molecule. In the myosin molecule, we have the head of the myosin molecule and the body of the myosin molecule. So this is one myosin molecule, and this whole thick filament constitutes of around 500 myosin molecule. In the head of the myosin molecule, this is the head of the myosin molecule, we have ATPase activity which occurs at the head of the myosin molecule. Now all of the myosin molecules, say suppose if this is the thick filament, right? The thick filament, we have the myosin which is arranged. Now the head is coming out, and whole of the myosin, body of the myosin is like that. So all the body are winded together like this. These are the heads and the body of the myosin. So this is how the myosin is placed in a thick filament. So if you see the structure again, if you go back to the diagram, then we have the myosin present like this in the thick filament. This is how the myosin is arranged in the thick filament. Then we have structure of the thin filament. So thin filament composed of actin, 7 actin molecules, 1 troponin molecule, and 1 tropomyosin molecule. Actin constitutes of the active site. So troponin and tropomyosin, they cover the active site. Now what happens, these myosin head are going to cling to the actin molecules which are present on the thin filament. So we have actin molecule which are present on the thin filament, and we have myosin heads that are present on the thick filament. So normally this myosin head, this actin is covered by troponin and tropomyosin. So it is covered by troponin and tropomyosin. When the muscle contraction takes place, there is uncovering of the troponin and tropomyosin, and this myosin head attaches or clings to the actin. And when it is attached to this actin, then the detachment requires ATP activity, and all the molecules, they get attached together and they get detached together. Then they click, say suppose if this one myosin head is attached to this actin filament, in once and it detaches and it attaches to the next actin filament. And this is how they are going to make the, so say suppose this third one myosin head is attached to the third actin, and then it attaches and it clings to the second actin, and this is how there is going to be contraction of the muscle. There is going to be coming of the Z line together in a sarcomere, and this is how the muscle contractions takes place. This is the real mechanism. So if you have seen a boat in Kerala, so this boat, the people, they row together. So if you have seen competitions of a rowing of a boat, so they row together. So all of these myosin head, they cling to the actin, they detaches, this detachment require ATP, and then they attach to the another actin. So this is how the contraction of the muscles takes place, and this phenomena is known as cross bridging cycling. So they attaches to each other, the thin filament active site interact with the myosin head. So we have 7 actin molecules which constitute of active site, which is covered by troponin and tropomyosin complex. Normally it get uncovered and then the active site becomes open. We have one molecule of troponin. So troponin, we have 3 kinds: troponin C, troponin T, troponin I. Troponin C, as the name suggests, it has affinity for calcium. Troponin T, it has affinity for tropomyosin, and I has got the affinity for actin. So I for actin. So the muscle contraction is because of the cross bridging cycling. We have 2 kinds, and during the contraction there are 2 terms. One is the tension, that is number of active cross bridges, and we also have shortening, that is spread of the cross bridge cycling. So shortening, tension is created, then there is shortening of the muscle. So tension is directly related to the number of the active cross bridges, and shortening is directly related to the spread of the cross bridge cycling. How much is the spread of cross bridge? If it is more, then the contraction is more. If the active sites are more, then the tension is more. So we have 2 terms, which is isometric contraction and metric contraction, as well as isotonic contraction. Now, as the name suggests, metric means we are talking about the length of the muscle. In case of isometric contraction, the length remains, iso means same and metric means length. So the length of the muscle remains same. There is only tension that is developed in the muscle in case of isometric contraction. In case of isotonic contraction, iso means same, tonic means we are talking about the tension. So in case of isotonic contraction, the tension remains same, and therefore the muscle shortening occurs. Work is done only during the isotonic contraction. And in case of isometric contraction, wherein the length remains same, no work is done. So there is no shortening, the length remains the same, only tension is developed in case of isometric contraction. So the skeletal muscle contraction, this is the walk along theory. The myosin, this is the myosin head. Now you can see it is reverted to the 45 degrees during the muscle contraction or muscle shortening. Now because of the release of the ADP, the myosin head detaches. You can see it is detached. So as I told you, detachment require ATP. So the release of ADP makes the myosin head tilted to 45 degrees, and then it binds to the calcium. So myosin head in presence of calcium oriented to the 90 degree, and it attaches to the another actin filament, the next one actin filament, and the cross bridge formation takes place. So in case of rigor mortis, wherein there is absence of ADP, in that case the myosin head remains attached to the actin at the same level. It doesn't detach because detachment requires ATP. So during the death, there is severe contraction of the muscle, which is known as rigor mortis. Now, in case of severe exercise muscle spasm, which is because of the accumulation of potassium, thin filaments are made up of actin, troponin and tropomyosin, while on the other hand, thick filament is made up of myosin. Relaxation protein is tropomyosin. In preload, there is isotonic contraction with a shortening of the muscle fiber and the tension remains same. The muscle blood supply increases during the exercise due to accumulation of active metabolites. Contraction of the covering binding sites on actin is prevented by troponin. So tropomyosin covers the myosin and prevents the attachment of actin and myosin. So tropomyosin makes a covering to the myosin, and that is why it is going to protect the attachment, and when it uncovers, then the muscle contraction takes place. So muscle contraction depends on the calcium. Duchenne muscle dystrophy is a disease of sarcolemmal protein. So sarcolemmal protein, we know it is dystrophin which attaches the thin filament to the sarcolemma. So this dystrophin protein, dystrophin gene defect is known as Duchenne muscular dystrophy. Skeletal muscle has the maximum oxygen consumption after liver, followed by the brain. Muscle phosphorylase is activated by calmodine, and calmodine and troponin both are the calcium binding proteins. So troponin also calcium binding protein. We have three components of troponin: troponin C, troponin T, troponin I. Troponin C has the affinity to bind to the calcium, troponin T affinity for binding to the tropomyosin, and I for the binding to actin.